and I'm a postdoc fellow at Massachusetts General Hospital, and I'm moderating today's section on how peripheral immunity influences CNS during aging and disease. So our first speaker today is Paras Minhas, who is a clinical fellow in the Department of Neurology at Stanford University School of Medicine. He completed his undergraduate at the University of Pittsburgh and finished his PhD training at Stanford University in Katrine Andreasen's lab. And he provided a fun fact that I have to embarrass him with, so I do apologize, Paras.
Okay.
So he gave it to us, so it's his fault, really. So from a young age, Paras has been terrible at sports, and when he was 10 years old, while attempting to play left center defender in his city league, he accidentally kicked the ball into his own team's goal. And so his teammates were not pleased, and it forever cemented his career choice into a non-athletic discipline. And we're very grateful for it. So hi, Paras.
Yeah, thanks.
All right. Our second speaker today is Aude Chiot, who is from France and completed her PhD in Séverine Boilet's lab at the Paris Brain Institute, where she studied how peripheral macrophages are implicated in amyotrophic lateral sclerosis. She is now a postdoc in Barea Jami's lab at OHSU, where she studies neuroinflammation and microglial subpopulations and their roles in various degenerative disorders. And she also provided a fun fact, so this is also on her. But near the institute where she completed her PhD, she unofficially adopted some cats that would come around when they ate outside, and she assigned them very scientific names, Einstein, Hawking, and Curing. And I'm assuming you did not bring them home with you, I hope. Okay. So welcome both of you, and thank you so much for joining. We ask that the audience please stay muted and to have your videos turn off throughout the webinar. And during the speaker's presentation, feel free to include any questions that you might have for them in the chat box. And then at the end of each of their talks, we'll open up the chat box and go through some questions, and they'll be able to answer those for you. So yeah, thank you again both for coming to present your work today, and I'm really looking forward to your talks. So I guess, Paras, the floor is yours first. So you can,
Okay.
I think you should be able to share your screen.
Yeah, so let's see if I can do that. And I'm wondering if it will work with, can you guys see the PowerPoint? Okay.
Yeah, it's all good.
Okay, great. All right, good morning, or I guess on the East Coast, good afternoon, or wherever you may be. I hope you're having a wonderful week. If not, I'm sorry to hear that. My name is Paras Minhas. I am a clinical fellow in Katrine Andreasen's lab in the Department of Neurology at Stanford. And today I wanted to share with you one of our recent stories that just recently got published on restoring sort of both central and peripheral immune metabolism and its effect on cognition and aging. So with that, I'll just get right into it. As I'm sure many of you on this call are aware, we are aging as a population. That is the number of older Americans from 1960, and that is to 2040, is actually projected to be over 1 in 5 here above the age of 65. And with that, comes a great degree of problems. So as we age, though, we actually find that our quality of life is not necessarily increasing. So when we progress from midlife, so 30 to 60 years of age, to mature adulthood, we start to get age-related diseases such as Alzheimer's or metabolic syndrome. And then as we go to late adulthood, we end up with more chronic diseases such as frailty, which leads to multi-morbidities such as hypertension and other such problems. And as such, the recent data actually released from the World Health Organization based in the United States here actually shows that while the average life expectancy is steadily increasing, though you can see it's starting to plateau, and I have no doubt that COVID has unfortunately negatively impacted this, the health span, that is the degree in which we live healthily with a period of without disease, has been relatively stagnant since the 60s and 70s. And you can see that's right around this mark in which many of us may have midlife crises or times are not looking so great. That is right around that age of 55 or so. And unfortunately, the measures and pharmaceutical interventions have done little to sort of increase this health span, which sort of brings us back to the drawing board here. And this is where sort of the Andreasen lab enters. And we sought to examine, what is going on in aging. So first off, aging is actually characterized really by an accumulation of these pro-inflammatory mediators, and whether it's in the periphery or in the CNS. And in particular, when we have states of stress and modulators of stress, they typically bind receptors, what we call pattern recognition receptors, on our innate immune cells. And these can include, macrophages, antigen-presenting cells, dendritic cells. And these then release further modulators that then can influence the CNS. And so in a normal, healthy human being who's relatively young, say in his or her 30s, these modulators tend to lead to quiescent brain activity and tend to be overall very healthy. But as we age, we actually accumulate what we know as damage-associated molecular patterns, or DAMPs. And these can come in many forms, but the predominant forms are things that are reminiscent of are byproducts of autophagy, such as cellular debris, mitochondrial DNA, and also reactive oxygen species, in addition to external stimuli, such as PAMPs, which are known as pathogen-associated molecular patterns. And these can come mainly from your microbiota or external infections, as well as environmental stimuli, such as, for instance, if we eat fast food or make unhealthy food choices, in addition to any sort of external pathogens that may enter or affect us as we age. And this, of course, is still going to go through the same innate immune system, and these then release pro-inflammatory mediators that then cause inflammation in the brain and lead to more adverse outcomes. And so, as I mentioned, aging is characterized by an accumulation of these responses. And as a result of that, when we see an accumulation of DAMPs, our once normal quiescent macrophages or microglia that are mainly anti-inflammatory. They exhibit homeostatic functions such as phagocytosis and exhibit immune homeostasis, actually tend to become more pro-inflammatory. And instead of surveying the tissue, they cause tissue destruction and release, of course, of reactive oxygen species. As a result of this, these quiescent myeloid cells then become largely activated myeloid cells. And so the question that we really had at the beginning was, what are some of the cell extrinsic modulators of aged macrophages, and how do they impact their function here? And so to study this, before my time in the Andreessen lab, there were two sort of studies here. The first was looking at the impact of NSAIDs. NSAIDs are your non-steroidal anti-inflammatory drugs that inhibit cyclooxygenase 1 and 2. They decrease PGE2 levels by inhibiting the main enzyme that metabolizes arachidonic acid to PGE2. And this was we showed that if you give chronic administration of NSAIDs to Alzheimer's and aged mice, it actually decreases their inflammation and they have better cognition. Of course, this is not very possible in a human setting because chronic NSAID administration is disastrous, especially in aged humans. It leads to complications such as gastric adenocarcinoma, as well as can also lead to chronic hypertension and renal failure. So this is really not an option, but this was sort of a first pass back about 5-6 years ago. And so a more nuanced pass was actually knocking out one of the EP2 receptors, the PGE2 receptors known as EP2 specifically in myeloid cells, so using a CD11B Cre line. And in this regard, we also found that this leads to beneficial outcomes such as decreased inflammation, as well as restoration of sort of these quiescent myeloid cells. But one thing that we also noticed, at least in the Alzheimer's models, was that there was a rescue of long-term memory. So when I entered Katrina Andreassen's lab, I was very interested in looking at, does this impact aging or not? And through what mechanisms? Both of these studies predominantly looked at as an outcome measure, but didn't necessarily go in depth into the mechanistic aspect of this. And so first thing I noticed is that when we collect peripheral blood from both young and aged donors, and we're lucky here at Stanford to have the Stanford Blood Center, so they have no shortage of blood drives in which they take individuals who are young, we define as less than 35 years old, as well as those that are aged, that is greater than 65 years old. And we measured in human monocyte-derived macrophages, their actual synthesis of prostaglandins through COX-2 and the subsequent downstream PGE2 synthase. We actually see that these are increased in human cells. But more importantly, what we find actually in the cells themselves, that they produce PGE2 to a much greater extent in the aged as compared to the young here. And so the question really was, does this have a functional impact in terms of the macrophage? And is this replicated in mice? Because of course we can't carry out these sorts of studies in humans, at least not yet. And so we took young and aged mice. The aged mice came from the NIA here. They were about 2 years of age compared to 2 to 4 month old. And when we look at their plasma and cortex, what we also found in vivo was that there was an increase in the PGE2 levels, both peripherally and centrally in the cortex here. And so one thing that we found that was interesting is that when we increase the levels of PGE2 in vitro, we actually find that they decrease both on the left here, the oxygen consumption rate, which is a marker really for your mitochondrial respiration, as well as the ECAR, which is more a marker for extracellular acidification or lactate production here. And this was a dose-dependent response. And similarly, when we take a specific EP2 agonist, this is butaprost in this case, we saw a very similar effect. I should mention PGE2 impacts about 4 different EP receptors, EP1, EP2, EP3, EP4. What I'm presenting to you today is obviously the snapshot of the study. We did look at the other EP receptors and didn't see any consequences of modulating those. But the one thing I want you to keep in mind for later on is that we see this sort of parallel decrease in both the OCAR as well as the ECAR, which is pretty unprecedented because usually the two sort of go reciprocal of one another. So this overall decrease in metabolism was surprising to us. Furthermore, we can reverse this if we actually use an inhibitor of EP2, and in this case was a Pfizer compound here known as PF044. This was actually not blood-brain barrier penetrant, as well as if we use a blood-brain barrier penetrant one from Amgen known as C52, both of them in a dose response manner actually rescued OCAR and ECAR here. And so the question becomes, does this actually impact immune profiling? And there's a lot of literature, especially from Luke O'Neill's group, as well as others, indicating that metabolism impacts immune function, especially in the context of innate immune cells. So we took young and aged wild type, as well as CD11B Cree EP2 knockdown mice, conducted immune profiling using Luminex, looking both at the periphery as well as the central nervous system here. And what we find actually is that the aged CD11B Cree, or these are really equivalent to aged wild type, have upregulation of the canonical inflammasomes such as interleukin 18, IL-1 beta, as well as some chemokines known such as MCP1 and MIP1 alpha. Similarly, we saw same profile in hippocampi as well, indicating that EP2 knockdown is sufficient to sort of restore immune homeostasis in vivo in these genetically altered mice. But what we then wanted to see was whether or not that actually impacts spatial memory. So we looked at the young and aged mice, and I just want to give you sort of 2 representative traces here. This is a Barnes maze. In brief, we shine bright lights on top of this maze, and the mice have to find the target quadrant and the target hole in which they can then go underneath the maze and hide in the dark. And what we find is that in the aged, which are basically surveying every single hole here, there's about 20 holes or so, the aged CD11B Cree with the EP2 knockdown actually stick to the target quadrant and eventually do make it into the hole much faster. And here's a quantification of that. You can see that the percent time in target quadrant is rescued here in the aged, as well as the distance traveled to target hole. They take a much more direct path, indicating that myeloid knockdown of EP2 is sufficient to rescue spatial memory in aged mice, okay? But of course, nobody is usually walking around with a floxed EP2 gene in them as a human being. So we wanted to see, are there pharmacological interventions that we can take here? And so could we inhibit this axis, the PGE2, EP2 axis, to normalize sort of the myeloid cell function and rescue cognition? And in brief, yes. So first we started with a blood-brain barrier penetrant EP2 antagonist, and we gave it in young as well as aged animals. These are now wild types. We gave it for approximately 1 month, so it's in 10 mg per kg per day. And we find that in the hippocampi, there's actually a restoration of the immune status. So on the right here, we can see upregulation again of pro-inflammatory cytokines, as well as downregulation of IL-13, which is largely considered to be anti-inflammatory. And then with the age C52, you can see that it sort of clusters together with the rest of the young cohort here. indicating that there is rescue of an inflammatory state in vivo in wild type mice when we give 1 month inhibition of the EP2 receptor. But then does that lead to cognitive rescue or not? Again, we see something very similar in both the novel object location. This is similar to novel object recognition, except that we just move the location to a novel part of the square, as well as on the right in Barnes maze here, we can see that they take a much more direct path towards the target hole. So we show so far, at least, and I should mention there's a lot more to this story than I have time to present, but I just wanted to give you sort of the main findings, that a C52 blood-brain barrier penetrant EP2 antagonist is able to rescue inflammation and cognition here. But the question really comes, why does inhibiting this actually reduce inflammation and aging? And so for that, we need to go to a little bit back down memory lane of basic biochemistry and look at how glucose is metabolized. So we're traditionally taught that there's this linear pathway. Glucose enters a cell, it undergoes phosphorylation either through glucokinase in the liver or hexokinase in peripheral cells, becomes glucose 6-phosphate, and then through the glycolytic pathway becomes pyruvate here. Pyruvate can then enter oxidative phosphorylation, and this is what we measure in a seahorse using OCAR, or it can enter extracellular acidification and becomes a lactate here. Turns out this is actually, of course, a very oversimplification. There are many side branches to this pathway, and today we'll be talking about one of them very briefly. It turns out glucose 6-phosphate can also contribute to a process known as glycogenesis, which is glycogen accumulation. It's largely thought that this process is limited to the kidney as well as liver. It turns out that we now also believe that it's found in macrophages. And actually, there was a recent paper, I think in 2017, 2018, at Cell Metabolism that also demonstrated a similar phenomenon in dendritic cells. So immune cells do accumulate glycogen as well. And it turns out that inhibition of the EP2 receptor actually prevents glycogenesis or glycogen synthesis. So when we profile the pathway, we know that PGE2 binds to this GPCR EP2. This then activates through serine 473 AKT, or protein kinase B. AKT then gives an inhibitory phosphorylation on GSK3 beta, effectively sequestering GSK3 beta. And GSK3 beta is actually known as glycogen synthase kinase 3 beta. And GSK3 beta's main role is actually to inactivate glycogen synthase. And so what ends up happening is when GSK3 beta is inhibited, glycogen synthase 1 is actually constitutively on. So we're effectively inhibiting the inhibitor with PGE2 stimulation, or EP2 agonism. And so what we get as an end result is glycogenesis or glycogen synthesis. And so if we take these immune cells and measure their glycogen levels, both with butaprost as well as C52, butaprost being an EP2 agonist, C52 being an EP2 antagonist, we can actually modulate the glycogen levels in these cells. Okay. So why does that actually lead to this immune phenotype and cognition that we're seeing? So turns out we wanted to see whether or not there is actually accumulation of glycogen that occurs. And so you can use a mass labeled glucose in which we label all the carbons with 13C. So it's just a heavy labeled isotope. And we looked at GYS1 function, and you can actually trace where the glucose goes in these human MDMs as well as mouse macrophages. And we see that in the vehicle aged, the blue here, there's a much more labeling, indicating that there's much more flux through the pathway into glycogen, versus in the traditional TCA cycle, that is oxidative phosphorylation, you can see that there is not as much labeling, less glucose is making its way into the TCA cycle. And that, of course, is reversed with C52 administration in the aged here. And so it turns out that we can rescue the metabolic profile with C52, and we believe for this reason. Now, one thing I didn't mention, I don't have time to show you, is why is this unique to aged? Well, it turns out that aged cells in particular are very, very glucose dependent. When we think of the three main macromolecules that come in and serve as a substrate for oxidative phosphorylation, they are typically your fatty acids, your amino acids, and glucose or glucose derivatives. Unfortunately, though, and again, unfortunately don't have time to show this data, the glucose is the predominant substrate that is preferred by the aged. the aged macrophages don't have the metabolic flexibility to modulate or to take in fatty acids and as well as amino acids in the presence of glucose depletion, or if they're being sequestered into glycogen, which we see in this case here. And so we're also wanted to know, is peripheral inhibition of EP2 sufficient? Though we demonstrate a similar phenomenon in microglia in vivo as well, we wanted to see, is it possible that if we peripherally inhibit EP2, do we restore immune homeostasis and cognition in the aged mice? And so here we use the blood-brain barrier impenetrant compounds. We gave it for 6 weeks. This is a Pfizer compound. Ran mass spec, we see for sure it does not enter the brain whatsoever. The reviewer actually one of the reviewers wanted us to really ensure that it wasn't in there by looking at the signaling cascade in isolated microglia. And for those of you who know about microglia isolation, getting enough microglia for a Western is really, really a large pain. But we did it and showed that as well. For whatever reason, he or she thought that this was not sufficient enough to show that it didn't get into the brain. So peripheral inhibition, though, what we do, and actually I was pretty surprised by these results myself, we actually do show a rescue in both the spatial learning as well as long-term memory here, which we measured through LTP. So this sort of brings back sort of to the main conclusions that we see here, which are, one, EP2 signaling increases with age, and particularly in the innate immune system. This signaling cascade actually modulates the effector functions and aging of macrophages through GSK3 beta and GYS1. And selective blockade of this receptor actually reverses central and systemic inflammation, and peripheral blockade of the EP2 receptor is sufficient to rescue cognitive deficits in aging, which was pretty surprising to us. And so here's sort of the working model that we have. As you age, you have tremendous amounts of EP2 that is releasing all this signaling, which we are actually now actively profiling to see which factors are modulating the systemic influence on the CNS. But it turns out that if you inhibit EP2 and sort of the pro-inflammatory effector functions of macrophages, they release less inflammatory cascades, and so your brain tends to be much more quiescent. And so I'm going to leave you with a very simple idea at the end of this. That is, as we age and we see accumulation of misfolded proteins, DAMPs, PAMPs, et cetera, we find that there's a decline in healthy immune metabolism and an increase in maladaptive immune responses. And the question that we still have yet to answer, but we are starting to, is can we modulate this immune metabolism to sort of mitigate these maladaptive immune responses? And it's looking promising, I can say. And so, of course, this work is not done in isolation. It actually really took an international team to do all of this. I encourage you, if you have time, to look at the paper in Nature. There's a lot more to the story that I can present in 20 minutes, but I just wanted to give you a main idea. But with that, I'll take any questions, and thank you so much for hosting this.
All right, great. Thank you so much for that great talk. And congratulations on a ridiculous amount of work into all the authors. It was a really great read. So I think Jerry typed in the chat box, but if any of the audience members want to ask Paris a question, you go ahead and write a message there, and we'll go ahead and answer those. But I guess while people are sort of marinating on all that data, I could ask a question. So I actually thought that the experiments with EP2 inhibitors in mice were very interesting, and I'm wondering, if you stop treatment or sort of did a washout period, do you think that the beneficial effects would be maintained, or they would sort of wear out over time? Or do you think you'd have to do like intermittent dosing to maintain those beneficial effects?
Yeah, that's a great question. And actually that's one that we're sort of exploring right now. What I can say is that based on sort of when we look at the cytoarchitecture of the brain, and specifically the microglia using TEM, we find that the mitochondria are repaired. Whether they stay that way or not without the EP2 antagonism is a question that we need to answer. I will say that the dosage that we gave, the 2.5 mg per kg of the peripheral EP2, that is the Pfizer compound PF044, is very low compared to what they were recommending using. But we did give it over a more chronic period. In short, I don't know the answer to your question. That is something that we need to explore, and I think that it is very promising, or at least it will be very interesting to see what happens in that case. Yeah.
So are these compounds like realistic to be administered to humans? I know nothing about the compounds, so I'm just wondering.
Yeah. So to my knowledge, these compounds come with some toxic side effects. We don't find them in mice. But I think they did run a phase 1, phase 2 trial in the context. PGE2 plays a large role in things like polycystic ovarian syndrome, as well as, a lot of the genital urinary syndromes. So I think they did find some adverse effects with that. So these two compounds in particular, I'm not so sure that they can actually be used. That being said, we are actively developing compounds to target these right now, the EP2 receptor, that have a much higher safety profile and much wider therapeutic index. Yeah.
Cool. So we have another question from our master host, Jerry. Do you have any guess what peripheral cell substrates might be responsible for this phenotype?
Yeah, so everybody really is focused on this TGF beta, as well as maybe eotaxin, CXCL10. My guess is it's not going to be just one factor. It's sort of hard for me to wrap my head around, given sort of these large changes that we're seeing on just one or two factors. I think it's more so a cassette of factors. And so my guess is more sort of the traditional inflammasomes, downregulating those, chronically probably plays a much better role in terms of rescuing brain tissue and ensuring that's healthy. So we're talking interleukin 18, IL-1 beta. As of right now, I don't have a clue as to which factors. Again, those studies are literally ongoing in the lab. And we hope to be able to answer that in the next year or so. But again, I'm doubtful that it's a single factor. I think it's going to be a whole cassette of factors that we're looking at.
And do you find that there are any anxiety-like behaviors after treatment in these mice?
Yeah, no, actually not at all. If anything, they tend to be a little bit more social. And one thing that we do notice is they seem to be a lot healthier too. So if anybody's ever received, mice from the NIA, or you age mice from Jackson, you know that they typically exhibit this metabolic syndrome. They're usually pretty chunky. They're not too healthy. These mice actually very slowly slimmed down and were much more active. So this is also another sort of active area of investigation in the lab, whether or not EP2 modulation plays a role in metabolic syndrome itself.
Yeah, so that was actually part 2 of my next question. I'm wondering what other type of age-related conditions could be benefited by inhibiting the PGE2 cascade.
Yeah, so I mean, at this point, I'm not sure. But what I can say is that since PGE2 plays such a large role in propagating inflammation, any one of these conditions such as metabolic syndrome, obesity, hypertension, a lot of these could possibly be targeted by modulating specifically the EP2 receptor. So if we think about the action of NSAIDs and how they decrease the concentrations of prostaglandins, that has beneficial effects, short term, right? So if anybody's sort of gotten recent vaccination from COVID shot, or, if you've gotten a headache, you take an NSAID, something like Tylenol, you feel better pretty quickly, right? The problem is, though, that a lot of these other EP receptors play very beneficial roles. So that's why we need to sort of hone the targeting specifically to more EP2 receptor in particular. And so if we follow sort of that traditional cascade, that if NSAIDs are having this beneficial effect by impacting all of the EP receptors, and we know that it's a general anti-inflammatory, one can imagine any condition that has tremendous inflammation and sort of chronic inflammation could potentially be modulated by the EP2 inhibition.
And I guess my last question, which was supposed to be the inverse, but I'm going backwards. Yeah. This is from my experience, because obviously I do Alzheimer's disease research. So the effects on cognitive function with respect to aging were really interesting to me. But there's sort of this inverse relationship between a person's risk for Alzheimer's and a person's risk for cancer.
Yeah.
And the theory is that those immune mechanisms that are responsible for, a robust response to cancer cells, so being able to patrol the body and then react to cancer cells, would otherwise impinge upon or converge on Alzheimer's disease pathways, which would increase a person's risk. So I'm wondering if you think that perhaps dampening this PG2 mechanism that would otherwise, totally hypothetically, maybe prevent someone from developing cancer cells, but might actually improve their cognitive function if you're basically making someone more susceptible to other age-related disease mechanisms while making them less susceptible to others, if that makes any sense.
Yeah, so basically you're asking, by dampening the immune response, is it possible we increase the risk for developing cancer or other malignancies?
Yeah, exactly, or other disease-related, yeah.
Yeah, it's definitely possible. That being said, what we typically see in cancer, the PGE2 receptor is largely, to our knowledge, localized on the innate immune response, and cancer modulation, and particularly the later stages of cancer, maybe not initially, is largely an adaptive immune response. So to our knowledge, this doesn't really impact the immune response to cancer, but then again, nobody's conducted these studies. So that's something that we would also need to look into as well. And in regards to the inverse relationship between Alzheimer's and cancer, I think that, yeah, you're spot on, and that there is this relationship. But one thing that I'd like to point out is, I think with the before the onset of Alzheimer's, right, that is sort of that MCI stage, there is a tremendous amount of inflammation that we see in patients. And so the question really becomes, if we intervene in that MCI stage, is it possible to sort of rescue the cognition Alzheimer's? I think that by the time we are clinically diagnosing Alzheimer's, it's a little bit too late, unfortunately, to intervene. So I guess where I'm coming from in terms of the metabolic modulation is, we would really need more of an earlier intervention when there's a large inflammatory component there. Yeah.
And obviously, I don't mean to put you on the spot for Alzheimer's. I know that's not what you were studying.
Yeah, I know.
Just related to the
No, I appreciate it. I mean, aging Alzheimer's related.
Yeah, exactly. And then we have a question from Angel. Hello, thanks for your lecture. What do you think about the link between EP2 and mitochondria?
Yeah, so that's I guess what I was sort of getting at, though I didn't get enough time to fully explain it. So what we find is that the EP2 sequesters glucose in the form of glycogen in these innate immune cells, and as a result of that, less substrate is able to make it to the mitochondria. And so I wasn't able to show you any of these, but we show with the TEM, as well as several different assays, that when we increase EP2 levels or PGE2 levels, the mitochondria tend to lose their beautiful cristae. There's a cristae malformation as well. And the reason, again, for that is that aged immune cells in particular are very glucose dependent. That is, they can't oxidize fatty acids as well as amino acids as well to sort of maintain that mitochondrial polarization necessary for oxidative phosphorylation.
Cool. All right, I think that should be it for the questions. Thank you so much for taking the time to answer all those.
Thank you.
Yeah. Oh, and I guess we'll